All right, everybody. Hello. Today we'll be talking about cell growth, division, and reproduction. And our learning objectives as we go through this whole thing generally revolve around one, what difficulties does the cell face as it increases in size? We're going to compare sexual versus asexual reproduction, and then we'll get into a little bit of cell cycle and mitosis stuff. And so just starting off basic, right? This is a little bit of math here. The surface area is the amount of covering the object has, right? And so if we're looking at this cube, it's the total amount of covering on the outside. Usually in a cube, that's length times width times 6. And the volume is the amount of space inside of that object, and that's length times width times height. So if we take this hypothetical situation where we have 1 by 1 by 1 cube, the surface area is 6 centimeters squared, while the volume is 1 centimeter cubed. But as you increase in size, as the cube grows bigger and bigger, the surface area and the volume are going to change at different rates, right? So let's just compare. Surface area for the 2 by 2 by 2 is 24. Volume is 8. This one is 54 and 27, the 3 by 3 by 3 cube. And so as that cube is getting bigger, if you look here, the volume is getting bigger at a higher rate than the surface area is, right? And so we have what's called a surface area to volume ratio. In this first cube, the surface area is 6, the volume is 1, so the surface area to volume ratio is 6 to 1. In this cube, the surface area is 24, and the volume is 8. 24 divided by 8 is 3. And so in this case, the surface area to volume ratio is 3 to 1. And in this last one, the surface area to volume ratio is 2 to 1. So as the cube gets bigger, the surface area to volume ratio gets smaller. That's important because when we talk about this, we need to think about cells, right? As cells grow bigger, too many demands are placed on the DNA, and basically think of it as a large city with not enough roads and streets, right? All these people congested in one area, and there's not a lot of ways in and out of that city. The cell works the same way. If the cell is too large, the surface area isn't big enough to support bringing in enough materials for that cell, right? Because the whole point of the cell membrane, or one of the main points of the cell membrane, is to bring materials into and out of the cell. And if the volume inside the cell is getting bigger, at a bigger rate than the surface area is getting big, then you're going to reach a point where the cell gets so big that it can't support its own growth. And that's what we're looking at here. That's what is triggered, or that's what triggers cell division. And cell division is basically the process of having one cell split into 2 daughter cells. Before that happens, the DNA needs to divide or replicate itself so that each one of these 2 cells has its own copy of the DNA. And then basically the whole point of cell division is to make sure that the cells stay small and that surface area to volume ratio is high, so that they can continue to bring materials in and support the inside of that cell. This process is actually related to asexual reproduction because a lot of forms of asexual reproduction just rely on cell division. And asexual reproduction is when one parent produces genetically identical offspring to itself. There's a few different ways. Bacteria, paramecium, they do what's called binary fission. You have things like flatworms, which can break half of its body apart, and it's called fragmentation. Sea stars do the same thing. Plants, you can do cuttings of plants and trimmings, and those can turn into copies of the adult plant. All of those are examples of asexual reproduction. Again, you can see binary fission, one cell splitting up into 2. It's pretty similar to mitosis, except it happens just in prokaryotic cells. Here is the budding I was talking about. This is called a hydra. This big long stalk that looks like a tree is the organism. And these little tiny nubs off the side is the growing bud that's the identical copy of it. So eventually this bud will get so big it just plops off and becomes its own thing. That's another form of asexual reproduction. Um, so that's different from sexual reproduction because sexual reproduction involves the fusion of two separate parent cells, right? You have one from mom, one from dad. They fuse together to make a genetically unique organism. While in asexual reproduction, you're making exact copies of something. So just comparison of the two really quickly. Asexual reproduction produces many offsprings in a short period because you don't need to find a mate. You don't need to dedicate as many resources to it. In stable environments, what you see is those organisms thrive, but oftentimes nature isn't full of stable environments for a long time. So if conditions change, those offspring aren't always well adapted. But in sexual reproduction, there's significantly less offspring. Growth takes more time. You need more time to find a mate. In changing environments, the genetic diversity is usually pretty beneficial. So that's our little basic overview. If you're thinking of the chapters in our book, that's 11.1. Now let's flip over to 11.2 and discuss the specifics of what happens when a cell divides, right? What's going on? So our learning objectives is we're going to talk about DNA and the chromosome, and then we're going to get into the cell cycle, describe the parts of mitosis, and then just investigate how daughter cells split apart at the very end. So in a prokaryotic cell, we'll get into eukaryotic cell in a second, but in a prokaryotic cell, the DNA is packaged in a single circular chromosome like this. And so when a cell divides prokaryotically, it just goes through binary fission, where the DNA replicates. So now I have two copies. The cell membrane splits, makes a little indentation, and now two identical cells are formed. Relatively easy, right? But in eukaryotic cells, which is what your body is made of, it's a little bit more complicated because we have long strands of DNA, very, very long strands of DNA that have to be compacted into much smaller packages so that they can be divided relatively easily. And so here's the double helix of DNA, right? That shape that's familiar that we should know. Those wrap around proteins known as histones to form these little packages called nucleosomes. And then those form these coils and supercoils, and eventually it forms this structure here called a chromosome. Technically, this is a duplicated chromosome where the left and right sides are identical copies of each other. We'll get to that in a little bit. And so looking closer at the chromosome here, you do need to know the parts. And so we have this part here made of chromatin. This is like the condensed DNA here. The single arm, right? So this side and this side are identical copies of each other. A single arm is called a chromatid. And together, if they're identical, they're called sister chromatids. In the middle, they're held together by a centromere. And then at the end of the chromosome, there are these structures called telomeres, which are really important in preventing aging and damage of the DNA. Okay, so again, the whole thing is a chromosome. Left and right side are identical copies of each other. They're called chromatids. Here's one chromatid, here's the other one. Centromere holds it together in the middle. Telomere is the cap on the end. Okay. I bring that up first so that you can understand what goes on in the cell cycle as we're going to talk about it here, right? So in a eukaryotic cell that is undergoing normal life sequences, there is this sequence of events called the cell cycle. And the cell cycle is split up into two major parts, and each one of those major parts is split up into smaller parts. So the first part we're going to talk about is interphase. That's the time spent in between cells dividing. Okay, it's made up of G1, S, and G2. The second phase is called the M phase, or the mitotic phase, which is made up of mitosis and cytokinesis. So you will see this diagram in a few different forms, but it's basically the same thing no matter which way you look at it, right? So this largest part of the cell cycle is called interphase. It's the time spent in between dividing. It's made up of 3 different phases: G1, which is basically cell growth; S phase, which is when the DNA starts to replicate, and then that leads into the preparation for mitosis. That's when things are constructed that are going to be used in cell division. And then cell division is the M phase, mitotic phase, that's made up of mitosis and cytokinesis. So let's look at each phase specifically, right? G1 is also called GAP1. That's just when the cell performs its normal functions. That's when it grows, it does cell things. If we're talking about a skin cell, it does what skin cells do. If we're talking about like a lung cell, it does what lung cells do. Then eventually that cell will grow so big that it can't keep supporting its own size. And so that triggers something in the cell that basically says, hey, it's time to start dividing. And so the next phase in the cell cycle is the DNA needs to replicate. And so in this part, the DNA replicates, which leads to then the cell moving into the last phase of interphase, where it grows certain things to prepare for mitosis, which is this last phase here. Okay, so synthesis is when the DNA gets copied. That's the S phase. GAP2 is preparation for division. Generally, the timing of the cell cycle is different for different types of cells. Certain cells go through the cell cycle relatively quick. The lining of your esophagus, small intestine, large intestine, those all divide within days, while like your red blood cells replace themselves every 4 months. While things like a smooth muscle is super long-lived. We're talking years here. Cardiac muscle is long-lived and it can't divide. So if you ever have heart damage, it can't repair itself. And then neurons, right? Neurons are long-lived and most of them don't divide. So if you ever have a neurological disorder where your neurons get damaged, whether it's the brain or spinal cord, those cells might not divide. And what we call cells that don't divide, technically they're in this little offshoot of the G1 phase called the G0 phase, where there's just growth, they live their life, but they're never going to go into the next phase here. Okay. So the next phase is called the M phase, or the mitotic phase, or cell division. Okay. That is made up of 2 distinct phases: mitosis and cytokinesis. Mitosis is when the DNA of the cell splits. Cytokinesis is when the cytoplasm and the rest of it splits. Okay. Mitosis is made up of 4 steps that you'll need to know. The steps go with an acronym called PMAT: prophase, metaphase, anaphase, telophase. PMAT. So take you quickly through each one. Prophase is when chromosomes are produced. That's how I remember it. That's when the DNA condenses into these chromosomes here, and you get these chromosomes produced. Prophase P, produce chromosomes. Metaphase is when the chromosomes line up in the middle of the cell. Again, middle M, metaphase. Next phase, anaphase. That is when the sister chromatids get pulled apart from each other, or they pull away from each other. Anaphase, chromatids get pulled away. Telophase, this is when 2 new nuclei are formed. Telophase, 2 nuclei formed. Cell's not all the way split, but the DNA is separated and it's forming its own nucleus now on each side of this larger cell. And that's mitosis, relatively simple, right? Prophase, chromosomes produced. Metaphase, they line up in the middle of the cell. Anaphase, they get pulled apart. Telophase, you get 2 new nuclei. That's mitosis. That's the splitting of the DNA. And then the final part of this process is its own separate offshoot is called cytokinesis. And cytokinesis is when the cell membrane pinches off, splits apart, and each one becomes its own cell. And that finishes the process here of the cell cycle. That finishes the process of the cell cycle. Looking back at our cell cycle, if I can go back here, that's the end, right? And then the new cells would go into the G1 phase. So The last thing we need to discuss is how this differentiates between plant and animal cells. In animal cells, the membrane just pinches off exactly like I described. But in plant cells, it doesn't split off as well. Instead, what happens is the cell plate forms, and eventually the cell plate will form a brand new cell wall that separates these two different cells.